So we're going to start looking now at all the different systems, our body symptoms, what is normal microbiota for that particular system, and then what diseases, what microbes can affect those particular systems, or what it generally affects them. So the skin, or the integumentary system, and the eyes are what we're going to be looking at first. So the skin and the eyes form a boundary between our internal environment and the external environment, or the outside world. The skin acts as the first line of defense, of course, against pathogens. All right, so if we're looking at the epidermis, we have different layers here. We have, of course, like I said, this is the integumentary system, and it forms the boundary between the human being and the environment, the organism and the environment. This is formed by the skin, the hair, the nails, and includes things like sweat glands, oil glands. The skin is our largest organ, and it covers the total surface area of 1.5 to 2 square meters. It's large. The thickness can vary from about 1.5 millimeters at the thinnest, which is in places like the eyelid, to 4 millimeters on the soles of the feet, palms of the hand. We have keratin, which is a protein in the skin, which helps give the cell the ability to withstand a certain amount of damage, things like abrasion, and help prevent desiccation, which is drying out. It also helps to keep water from coming in as well. So we have millions of skin cells that are going to be replaced, and so they're going to slough off daily and shed, and as they are sloughing off, they're going to shed off microorganisms along with them. So we have that process of keratinization, which you can see here. In the stratum corneum at the very top, you're going to have these cells, remember, that are going to be pushed up. They're getting further away from the blood supply down here, and as they're pushed up, they're going to be keratinized. So they're going to have keratin in them. They're going to form a thick outer layer, and the top layer comes off. All right. So it's a thick layer of epithelial cells, and that layer is going to be the stratum corneum. All right, so there's 5 layers here of the epidermis, and the one that we're most concerned with is that stratum corneum, because that is the layer that we shed off, and that's about 25 cells thick in general. That's a very broad generalization there. So these are dead. Like I said, they've migrated from those deeper layers, and then as they do, they get kind of squished together and form a nice thick layer, all right, of dead skin cells. It's protective in nature. And so then below the stratum corneum, we have 3 or 4 more layers of epithelial cells, depending on the location, total of 5 in the thickest places and 4 in most other places. The lowest layer is the stratum basale, or basal layer, basement, think lower, bottom layer. It's attached to the epidermis underneath, and this is where all those cells come from that make up the epidermis, okay? The dermis then is underneath the epidermis, epi meaning above, and it is composed of connective tissue instead of epithelium, all right? And so it's going to have a rich matrix of fibroblast cells and fibers like collagen, elastin. It's going to have macrophages and mast cells. It's also going to harbor a very dense network of blood vessels, which we don't have in the epithelium, nerves, and some lymphatic vessels. And so your root hairs, which are called follicles, are going to be in the dermis. We're going to have our sebaceous oil glands. Let's see, I'll show you that follicle over here. So here is the, here we go. Here is the follicle down here. All right. Here is your sebaceous oil gland. This is your a sweat gland here. The red and green here are your arteries and veins. All right. And then the yellow here is going to be, I believe that's supposed to be your nerves there. Okay. All right, and so you can see this erector pili muscle. Yeah, that's nerves there. Okay. All right, and then you have some adipose tissue, which is fat, basically. Okay, so we have, of course, I said these sweat glands. Let me get this little thing off here. It's not going to come off. Sorry, I'm having a little mouse trouble. So what type of defenses does the skin have? All right. So we already talked about that keratinized layer. That's actually a physical barrier type of feature there. So a protective feature is that keratinized surface. Then we also have our resident microbiota. Those are going to be part of our defense mechanisms. We have antimicrobial peptides, lysosomes. We have sebum and sweat glands. So antimicrobial peptides or proteins are going to be little positive chemicals or protein chemicals that act by disrupting those negatively charged layers of the membranes of certain bacteria. Okay, we have many different types of these particular peptides. These are responsible for keeping the microbial count on the skin fairly low. We also have sebum, which is going to lower the pH and make the skin inhospitable to microorganisms because the pH may not be right for them. It's also oily due to the high concentration of lipids that are produced. Those serve as nutrients for the normal microbiota. Normal microbiota, remember, are a defense mechanism because they take up food and housing, so to speak. And so they're acting via competition or antagonism. They also sometimes produce different chemicals or toxic byproducts via their metabolism that can inhibit the growth of microorganisms that are not necessarily adapted to the skin. All right. Sweat is going to be inhibitory to microorganisms because it's got a low pH and a very high salt concentration. Lysosomes are going to be found in sweat, but also in tears and saliva. And these actually can help to break down the peptidoglycan found in the cell walls of bacteria. So in order to survive on the skin, an organism, a microbe, must be able to survive in dry and salty conditions, okay? So they're going to be sparsely distributed, the normal microbiota, over flat areas of the body like the back. Okay, and they're going to be much denser, or they're going to kind of accumulate more in moist areas and skin folds such as the underarm and groin. And so they also they can live in hair follicles or glandular ducts depending on the microbe. We have hundreds of species of microbes. Some are actually well-known pathogens, but their different species are going to be found in different areas of the body, and different people can even have different species. So an individual's normal microbiota or normal flora is the old language, but you will still hear that, is going to remain fairly constant over time. And so you're going to see species here like Streptococcus, Staphylococcus, Corynebacterium, Lactobacillus, Pseudomonas, yeast like Candida, Propionibacterium, many different things. And why do we care? Why do we want to know which normal microbiota are there? Well, let's say you take a swab of the skin and you get 3 different bacteria. You cannot immediately assume that that is an infection or an abnormal state, right? Because some of those are going to be normal. So if you don't know what is normal, you don't know what is abnormal. So it's very important that we know what are the normal microbiota for this particular area. And remember that sometimes these are going to be opportunistic pathogens, meaning that when they get in the wrong place, or if something changes that mix and suddenly there's more food available, they may overgrow and become a problem. So as long as they're in their proper place in the proper amount, then they are helpful to us. All right, so looking now at some of the harmful bacteria. We said that Staphylococcus can be a normal microbiota. Well, what about MRSA? Everybody has heard of MRSA, right? MRSA is methicillin-resistant Staphylococcus aureus. Methicillin is an antibiotic, of course, and the Staphylococcus aureus species is a gram-positive cocci that has developed the capacity to be resistant to the last resort antibiotic, methicillin. Okay, we also have, and when I say last resort antibiotic, I mean when things are really bad, you want to go to and use this really strong antibiotic, methicillin. It's kind of like the last resort. We also have vancomycin as a last resort antibiotic, and we now have bursa or some vancomycin-resistant Staphylococcus aureus species. Okay, so Staphylococcus do not, all of them, none of them, I should say, have spores. They do not have spores. And so you can differentiate from Streptococci because because they produce the catalase enzyme, all right? And so this is just some little helpful tips here. So Staph aureus can cause meningitis. It can cause furnicles or carbuncles, which are just different types of these lesions and pustules here. Impetigo. and scalded skin syndrome, SSS, okay? These actually can remain viable after they dry out in the air. And they resist the effects not of just antibiotics, but also of some disinfectants. Very problematic here. Basically, any penicillin derivative or any medication made from penicillin, they can have the resistance gene, okay? And so you see these, we have our community acquired MRSA, and then you have hospital acquired MRSA. And so you do see this picked up, and those are just two different strains. You're more likely to get the hospital acquired in the hospital, so you see these picked up. But then you have the community acquired, and that's where you're more likely to get that particular strain is out in the community. Like, for instance, we had an outbreak, oh gosh, when my son was in high school, so 15 years ago, something like that, maybe even 20, it might have been junior high, and there was just a huge outbreak of MRSA among schools. And so it was during football teams, football season, and they would have pads and towels and things, and so it was spread that way. And so these are gram-positive cocci. They do cluster. All right, so that's where the staph, remember staph means clusters, kind of like grapes, little clusters of grapes is what this looks like. It can be very, very virulent, but it is, Staphylococcus aureus can be normal microbiota, okay? So really important to know this one. All right, so here we have some pictures. One of these is actually a brown recluse bite, and one of these is a MRSA infection, okay? These are very commonly I don't want to say misdiagnosed because it's mistaken, I should say, often between the two. Spider bites and MRSA bites look very, very similar, especially at the beginning of onset. So which do you think is which? Hard to tell, right? So the top here is going to be brown recluse and the bottom is going to be MRSA, okay? So you've got, well, the only prevention for MRSA is going to be very good hygiene, okay? So the signs and symptoms here are going to be, you're going to have a red, raised, very tender, localized lesion. It will often pit, which you can kind of see pitting going on up here, but it also will happen here. And it will occur very easily where you have an opening in the skin, so where that first line of defense has already been broken. And so fever can occur. So fever, generalized fever, but it's definitely going to be very hot at the site of the sore, okay? So transmission or prevention is only possible, like I said, with good hygiene, but transmission or treatment is going to be, they're going to want to incise that and get the fluid out, okay? Not squeezing, because that often drives things further in. You want to go and get this treated appropriately. And then sometimes they will treat with an antibiotic like vancomycin, okay? And so you can pick this up on surfaces, and like I said, gym equipment, which is where we had the trouble with all the high school students, airplane tray tables, razors, okay? So if somebody has MRSA, they need to keep it covered. They do not need to be out spreading that, all right? So MRSA is so virulent because it can produce lots of enzymes that helps it gain entry into the body. We can produce hyaluronidase, which will dissolve the hyaluronic acid, which can bind host tissues together, so that allows it entry. Staphylokinase, which digests blood clots. If you have an injury, one of the defense mechanisms is not just to stop blood making a clot, it's also to prevent entry of things. And so if you dissolve that little wall of a clot down, you can get inside. We have nuclease or DNase, that is going to digest DNA. Lipases also can help bacteria colonize these skin surfaces, okay? So we have polymerase chain reaction that we can use to diagnose. We can cultivate it on blood agar and look for the lysis of the blood cells. Mannitol agar can also be used if you have a very heavily contaminated species. Catalase can be used to differentiate staphylococci, which does produce it, from streptococci, which does not, and that's catalase is going to be used to break down hydrogen peroxide, H2O2. Okay, and so here we have a staphylococci coagulase test down here. At the bottom. Coagulase is that enzyme that reacts with factors present in plasma to start clot formation. So in the coagulase test here, we have a tube of plasma that is going to be inoculated here with the bacterium. If it remains liquid, it's negative, and if it develops a lump down here or becomes completely clotted, then it is positive, showing that it does produce that coagulase. All right. And here at the top, this is a blood agar plate growing Staphylococcus aureus. Some strains show 2 zones of hemolysis caused by 2 different hemolysins, and so here the inner zone is going to be clear. All right, and then here is the outer zone. Okay, so inner zone, outer zone. The inner zone is clear, where the outer zone is kind of fuzzy, and it appears only as if the plate has been refrigerated after growth. So as we're going through all of these bacteria, you need to make yourself flashcards, and some of these you will see in other particular organ systems, and when they come back up, I want you to know all of the information from the previous chapters. And so the best way to do this is to make flashcards. You can use this, you'll see on your study guides, it's in a table format, but if you make the flashcards, just send me pictures of those, okay? You can put like 10 in 2 columns, and you know, like an 8 by 10 sheet of paper size, and then take a picture of that and send that to me with your study guide, because that's going to be the bulk of your study guides for the rest of the units. Okay, and so you need to know MRSA is methicillin-resistant Staphylococcus aureus. You need to know that it is gram-positive, and you'll see they have these tables throughout the rest of the chapters. These are really, really helpful. Okay, very concise information. You can see here that it is coded as a bacteria. You could do color coding for this and say do all of bacteria a certain color, all viruses a certain color. Really, the way you do the color coding doesn't matter. You just need to have a purpose for it. Don't do just different colors. You got to have a reason for the color. You could put the name in one color. You could put the virulence factors in another. Okay. And now you don't always need all of this information on here, but you definitely need to know specific things. The name of the organism. You also need to know the name of the disease it causes. And I see students mix these up. Please don't do that. Okay. You need to know that it has superantigen and coagulase for sure. Let's see. Gram-positive, we already said. Treatment, vancomycin. MRSA, you probably need to know more of the epidemiology, like that we have community acquired and we have hospital acquired. And you need to know the organ system it generally affects, if it is a normal microbiota or not, that type of thing. Okay. A lot of times too, we may put if it has a capsule or endospore, so I would put no on these, and I would write with what it does not have as well. Like if I specifically point it out, you need to be sure and put it on there. Okay. All right, so all of our rashes have these different names, whether it is a pustule or papule. And so this is a maculopapular rash, okay? And so that is a flattish red type eruption here, slightly raised, but just still keeping that flat line above it. It might come up a little bit, but it's not going to be like a big bump, all right? And so some of the diseases that you will see that have this maculopapular rash are going to be things like measles, rubella, rubeola, Fifth disease, roseola, a lot of these childhood type diseases. And so understanding the differences in the rash is very important diagnostically. All right, so measles is rubeola. About 367 children actually die of this disease every day, even though we have a very effective vaccine. We've had it since 1963. You're going to see this in your MMR, measles, mumps, and rubella vaccines. It's really important to understand how vaccines work and to be able to explain to patients why it is so important to get our vaccinations. Okay, so you probably hear me harp about that quite a bit as we go through these particular diseases. Okay, we haven't seen some of the really very like terrible diseases like smallpox and all that we used to have. We just don't have those anymore because we have such good vaccines. Okay, before the vaccination, measles killed about 6 million people a year. That's a lot. Okay, that's a lot of people. And so since 2002, the deaths from this have dropped by about 74%. And the reason we even still have some is in developed countries like the US, we've got these anti-vaccination movements. We've got these people that are deciding not to vaccinate their children, and so we're losing our herd immunity, which is a big problem because herd immunity is how we protect those vulnerable populations. There's also been a very unfounded link between vaccines and autism, okay? And that has actually been proven to be not a causation. It is a correlation. I believe I've talked about this already, okay? So, rubella. Human beings are the only reservoir. The signs and symptoms are going to be a sore throat, dry cough, headache, conjunctivitis, and of course, this red maculopapular rash, okay? And so you can also have secondary infections of otitis media, which is an ear infection, sinusitis, subacute sclerosing panencephalitis can occur, SSPE, all right? So that's a serious secondary infection there. That we really need to pay attention to, okay? You can also have these little spots in your mouth called Koplik's spots, okay? And so that's another thing to look for. All right, so sequelae is a word that you will occasionally see, and basically that just means the consequences, okay? So when you see sequelae, it's just a fancy way of saying consequences, all right? And this is going to be consequences of disease or injury, all right? So a small percent of cases do develop laryngitis, bronchopneumonia, or those secondary infections, the bacterial sinus infections, okay? 10% can progress to pneumonia or encephalitis, okay? 1 in 1,000 of those develop subacute sclerosing panencephalitis, okay? And that's a progressive neurological degeneration of your cerebral cortex, which is the white matter of your brainstem, and in your brain, not just your brainstem, but in your brainstem as well. And this involves a kind of a defective virus that doesn't form a capsid and it doesn't get released from the infective cell. And so it spreads unchecked through the brain by cell fusion. It destroys the neurons and accessory cells, and it breaks down the myelin sheath. And so measles can also cause a very profound intellectual and neurologic impairment as well. It can lead to coma and death because of the central nervous system disruptions. All right, so what are the virulence factors here? The virus can actually implant on the respiratory mucosa, so it's got a respiratory portal of entry, and infects the trachea and bronchial cells. It can then travel onto the lymphatic system, multiply, and then enter the bloodstream. Once something enters the virus, once a virus enters the bloodstream, this is known as viremia. All right, and that can carry the virus to the skin and various organs. The cell membranes of host cells nearby can fuse into what's called a large syncytia, and that means one giant cell with many multiple nuclei, and so then they're no longer able to perform their functioning, okay? So how is it spread? This is one of the most contagious infectious diseases. Its transmission is by droplet, so it's respiratory droplets. Humans are the only reservoir, so we should be able to eradicate this disease if we can just get proper vaccination. So a person is going to be infectious or be able to spread this during incubation period, the prodromal phase, and the skin rash. But once they reach convalescence, they are no longer contagious, okay? And so how do we diagnose this? You can use the clinical presentation, look at that maculopapular rash, look at signs and symptoms. You can also use ELISA and test for the IgM antibodies for the measles antigen. You can, of course, prevent this. What is the best treatment? Is prevention. The prevention is going to be that MMR vaccine. This is a live attenuated vaccination, and so this will give you protection for up to 20 years. So you do have to get updates on this. This is recommended for healthy children aged 12 to 15 months, and then you get your boosters after that, especially when the child enters school. And so treatment, you're going to, of course, do some palliative care in reducing the fever, suppressing the cough, replacing the lost fluid. And then you want to relieve neurological and respiratory symptoms, keep electrolytes balanced, and so you can supplement with vitamin A as well. All right, next we have rubella. This is what's also called German measles. So that would be the common name, and that means little red. And so this is minor, this has a minor rash with a few complications, but the biggest problem here with rubella is it can cause congenital issues. It can cause serious damage to a fetus in utero, okay? So women of childbearing age must be vaccinated, okay? Very, very important. Actually before conception. And so if you get it postnatal, you will have a rash of pink flat macules and some papules, so maculopapular rash. It's usually going to appear on the face first and then move down the trunk towards the extremities, and kind of in that order. And this will advance or keep moving down and then resolve within about 3 days. Milder than the measles rash. Adult rubella, you may also see some inflammation in the joints instead of the rash. And again, the best prevention here is going to be what? Vaccination. So when rubella is contracted by a pregnant woman, it can cause congenital issues. It's teratogenic, okay? That means it's going to cause serious or teratogenic, serious effects. It could actually cause miscarriage, permanent defects to the child, okay? Most common is deafness. It can also cause heart problems with cardiac abnormalities, ocular lesions, deafness, physical and mental retardation in varying different combinations, okay? And so it also, and this is especially problematic in that first trimester. All right, so very, very important that pregnant women have had their MMR shot. Other consequences can be things like having anemia or hepatitis, pneumonia, carditis, and infections of the bones. So not good for babies. So rubella is from the ruby virus. This is togaviridae. It is able to stop cellular mitosis, which is, of course, a very important process for that developing embryo and fetus, especially in that first trimester. And this can cause apoptosis of normal tissue cells, which means apoptosis is programmed cell death. So if it causes that to occur when it's not supposed to, it can stop the progression and formation of organs. Okay, so it can cause really severe irreversible organ damage. It causes a vascular damage to your vascular endothelium, which can cause, again, poor development of organs. Okay. So to culture this, this mimics many other diseases, so this isn't going to be diagnosed just based on clinical grounds, okay? So you can use an IgM antibody test through ELISA or some latex agglutination tests. Prevention is treatment, MMR at 12 to 15 months, and then a booster at 4 to 6 years old, okay? Postnatal rubella is generally benign and requires symptomatic treatment, okay? So just palliative care, treating the symptoms. No specific treatment for congenital rubella. Best treatment is prevention. I just cannot say that enough, how important these vaccinations are. So this is going to be distributed, of course, across the world. It is going to be spread via contact and respiratory secretions, occasionally urine. The virus is shed during the prodromal phase and up to a week after the rash appears, okay? Infants infected congenitally are going to be contagious for a much longer time. Virus is only going to be moderately communicable. If you have very close living conditions, then that's going to allow it to spread. We eliminated this in the U.S. in 2004, in both Americas in 2015. I do have some fears that we could see resurgence of some of these things because we've already seen resurgence of other things. partially because of the anti-vax movement. Some of it is going to be due to alternative strains, mutations coming out, but the anti-vaccination movement is really putting many people in harm's way. So Fifth disease is the common name for another childhood illness. This is arrhythmia infectiosum. This is caused by parvovirus B19. It's going to cause a febrile disease with a bright red rash on the cheeks. There is no vaccination for this. You have a very slapped cheek appearance with this. It's going to have a maculopapular rash lasting a few days to weeks. All right. And so, like I said, parvovirus B19, you can usually diagnose this via clinical presentation, but it is important to rule out rubella by testing for, or you can rule it out by testing for IgM antibodies. This is very contagious. You'll see this run through things like daycare centers. This is a very mild disease, and so it's mostly palliative care. You're just treating the symptoms, okay? There's no vaccination for this either. It's just not necessary. Next, we have ROSOLA, all right? This is human herpes virus number 6. You're going to need to be very careful with the herpes viruses. We have a lot of them, okay? And so 6 is what this one is. And so you'll need to really find a way to keep those separated for yourself. This is like another common childhood illness. You get this maculopapular rash, high fever. It can be up to 3 days. If the fever is high enough, you may actually see some seizures with this, which is problematic. It can actually remain latent or sit on the host indefinitely after the disease has cleared away, and then reactivate in childhood or even adulthood. And then you're going to have mononucleosis-like or hepatitis-like symptoms with that. It is thought about 100% of the U.S. population has been infected with this virus by adulthood. There's no vaccine and really no treatment. It is palliative care. All right, so this chart is in your book of these maculopapular rashes. And so this is kind of, there are some other things you can look at that can cause this besides these. It's going to be Zika virus that we will look at, scarlet fever, secondary syphilis, and Rocky Mountain spotted fever are some that are not listed here. So you can use these to kind of differentiate. These are the 4 viruses we looked at. This is a really good slide to make flashcards off of. And so Fifth disease is the common name of, or the name of the disease that you get. The virus name is parvovirus B19, and then there's your human herpes virus 6 causing ROSOLA, your measles virus causing measles or rubella, and then we have rubella virus, which also causes rubella, okay? And you can kind of look and compare and contrast the differences here, and we talked about most of these. So let's wrap this up with another NCLEX prep practice question. We have a pediatric client that presents to the office with ruddy or reddish cheeks and a low-grade fever of 101. So the client's mother states that the child has been acting normally, but he noticed the child has a slight rash, mainly on the torso. Which disease do you suspect based on the clinical presentation? All right, so hopefully you pause this and have come up with your answer. When you see that slapped cheek appearance and a not very sick child, just a low-grade fever, that's often going to be Fifth disease. So this is B, Fifth disease, not very sick, slapped cheek appearance.